Hello, my name is Antonio Mendez. I'm a student at Cal Poly in the Plant Science Department, and I did my scientific poster on benefits of native mycorrhizal amendments to perennial agroecosystems increases with field inoculation density. In traditional intensive or industrial agricultural cropping systems, the community of arbuscular mycorrhizal fungi are usually destroyed by anthropogenic inputs such as tillage, fertilizer, or pesticide application. Whereas perennial polyculture cropping systems provide ecological benefits such as carbon and nitrogen sequestration, increased soil physical and chemical properties, and an overall reduction of anthropogenic inputs by mirroring some native perennial ecosystems. This study shows that native arbuscular mycorrhizal communities can increase crop growth and survivability depending on crop species and inoculum density. Also, current commercial arbuscular mycorrhizal fungi inoculum rate recommendation and species selection prove ineffective in enhancing crop growth and survivability, while suggesting alternative distribution methods may be necessary to utilize native AMF as an amendment in agroecological systems. As we know, polycultures can improve and provide sustainable agroecosystem health and crop production. Arbuscular mycorrhizal fungi association with some crop species can improve soil nutrient assimilation and distribution and can alleviate abiotic and biotic stressors such as drought, herbivory, and pathogenic infection. This study tests the effectiveness of native AM fungi in increasing plant growth and survival on cereal, oilseed, and leguminous crops. Commercial mycorrhizal inoculum is usually non-native to the application area and is recommended at low rates, proving ineffective. It remains to be seen whether the ineffectiveness of commercial mycorrhizal stems from low application rates or the non-native inoculum being maladapted to the introduced soil or water regimes where they are applied. Both greenhouse and field condition experiments were conducted to test if native AMF inoculate enhanced crop survivability and growth. Using 19 crop species with production potential, both experiments looked to determine plant response to inoculation using various crop species, inoculum densities, and rates, as well as growing conditions. The field experiment was analyzed by taking the growth and a total number of surviving plants annually to determine the plant's response linked to the rate and density of inoculum applied. For the greenhouse experiment, PROC-GLM or PROC-GLM was used to determine the dry weight biomass of each plant under different inoculation rates and densities to determine plant response to arbuscular mycorrhizal fungi. In field conditions, plant survival varied depending on species, inoculation rates, and densities. Across all species, commercial AM fungi recommended inoculation rates were not great enough to produce a significant enough effect to increase survival. Some species growth depended on inoculation, while other plants were not responsive to native AM fungi or suffered deleterious effects. In the greenhouse, 19 plant species grew 14% larger with the inoculation of AM fungi compared with those non-inoculated. However, growth varied significantly across species. The top 8 plants experiencing the most growth were leguminous, while those experiencing neutral or decreased growth and survival with an increase in AM association were primarily C4 grass plants. Going down to the center of the poster, we can see figure 1, showing the percent increase in plant growth due to native mycorrhizal inoculation. And as the above text suggested, the first 8 showing the most growth in response to AM symbiosis are leguminous. Going down to figure 2, figure 2 shows that the mycorrhizal enhancement of plant growth on individual plant species in the greenhouse was strongly correlated with the response of the plant species to inoculation density in the field. And as the text suggests, on the x-axis, we have the mycorrhizal response in the greenhouse. And on the y-axis, we had the response to the field. And our y-axis corresponds with increasing inoculation density. In figure 3, we have the mycorrhizal enhancement of plant growth on individual plant species in the greenhouse, being strongly correlated with the response of the plant species to inoculation density in the field. Very similar. to the adjacent graph. However, it focuses primarily on growth. And it's using the same X and Y axis as the inoculation rate increases, plant growth increases. The studies show that AM can increase the productivity and survival of leguminous crop species without significant adverse effects on most non-mycorrhizal species. Plant responses to inoculation and non-inoculation treatment in greenhouse and field conditions were correlated, which hints at a replicability in real-world environments. The inactive commercial inoculation of non-native AM may result from the introduced fungi being unable to adapt to the soil's new nutrient, microbe, and water conditions, as well as commercial application rate recommendations being of a much lower density than native mycorrhizal amendments used in other scientific studies. However, such densities may be cost prohibitive. This research highlights the value of allowing the soil microbiome to perform its natural function in agroecosystems. It shows that arbuscular mycorrhizal fungi communities are capable of increasing yield and production crop species significantly. Using a native inoculum of beneficial microbes as an amendment to planting compared to commercial inoculum or non-native inoculum, or non-inoculation can improve plant productivity and establishment. My name is Antonio Mendez. This has been my scientific poster for crop ecology. Thank you.